Good morning, everyone. My name is Pauline Cervantes, and this summer I've been looking at the benefits of two beneficial endophytes on the growth as well as the endomycobiome in the host, which is meadow fescue. So this project is an extension of Lian Nguyen's PhD chapter, which looked into Metarhizium, novozealandicum, C14, and epicloe endophytes and their activity against grass grub herbivory. And so, although we weren't able to replicate the study because it's the wrong time of year and we didn't have grass grub, we still wanted to look at how C14 and the epicloe were able to manipulate the endomycobiome as well as the growth, and whether this was possible to replicate for my samples. And we also wanted to look at the antimicrobial activity of Metarhizium novozealandicum against the fungal species that we were able to isolate from meadow fescue. So initially what we had was I'd grown the sward of 5 plants from the samples of grass that we had. We had 4 different species. We had 4 different species of epicloe. 2 of the plants had the same, the U2 epicloe, as well as the EM142 and 1 lithol. And what we did was then we harvested the plants after 2 weeks. After 4 weeks, sorry, we inoculated the plants with C14 spore suspension after 2 weeks of growth, and after another 2 weeks, we then harvested this. And one of the 5 swards was then surface sterilized and then plated onto Metarhizium selective media. So what we found in the plant dry weight, which is that we didn't find a significant difference between the Metarhizium treated and the control plants. So, and in order to detect such small effect, we might have needed to increase the sample size. And after the surface sterilization of the plant samples, we then calculated the colony forming units, and we enumerated this from the plates themselves, from the leaf, and from the roots. And afterwards, what we found was that the plants with Metarhizium inoculated in them generally had more CFU counts, as you can see from the top right corner, except for the ones that had the EM142 epicloe strain. And afterwards, we then extracted the DNA, amplified and sequenced the ITS region using the primers ITS4 and ITS5. And we then found that generally the ones with the lowest species richness had no epicloe and no Metarhizium, but these are just the tentative results because the study is still ongoing and we're still sequencing more DNA. So this is a more holistic summary of the fungal species that we found. So we found quite a few isolates that were only isolated once, but there were a few that were isolated multiple times from both the control and the ones that were treated with Metarhizium. And this subset of fungi could potentially be the core endomycobiome of meadow fescue. These fungal species have also been commonly extracted from other grasses such as cocksfoot grass, Yorkshire fog, as well as red fescue. And we also found other species that were only present in Metarhizium and ones that were only present in the control, for example, Purpureocilium and species of Penicillium in the control. However, we were only able to retrieve 2.5% of Metarhizium from the plants that were inoculated. And this is basically 1 out of 40 plants we were only able to get Metarhizium from them. But we were still interested to see of the effect of Metarhizium, whether or not the fungal species we were observing were just by chance or whether or not it was actually Metarhizium in action. So we still went ahead with the dual culture analysis, and so far it's still ongoing, but what we found was that we've seen a slight inhibition by C14 against trichoderma, and the other 14 much more slower growing species is still yet to be quantified. And so far we've identified 56 different isolates, and 19 of which I've only recently gotten back. So I'm still yet to analyze those, but what I'm hoping to get with the data is that once I have all the fungal isolates identified, tracing this back into the ones that were controlled and ones that were treated with Metarhizium, and looking at the incidences from the different breeding lines, and from that we're able to gain a better understanding of how the endophytic microbial communities have actually been affected. And so watch this space, there's still a bit more to be discovered. Thanks for listening, and thank you to my supervisors as well.
Thank you. Thanks, Pauline. Questions for Pauline? I have a question. You had a lot of the fungi there looked like they were pathogenic, Fusarium oxysporum.
Yeah.
So obviously the Metarhizium didn't do much against the pathogens.
Yes. So we have different species of Fusarium that we were able to isolate. Which the thing is, it's sort of hard to tell how effective the Metarhizium really was, and to be able to tell what Fusarium actually does in the plant. And I guess it's the dual culture analysis will show that. We've seen one species, it's currently growing at the moment, I can't be too sure, but there were some species of Fusarium that outgrew the Metarhizium, but then there was one species of Fusarium, Solani, its growth was much more retracted compared to the other species of Fusarium. So hopefully that will be more clear with the dual culture analysis. But yeah.
Other questions?
Whereabouts does Metarhizium end up within the plant?
Usually people find it in the roots, but so far where I found it is in the leaf region of the plant, and where I've isolated it, it's only Metarhizium that was present there, no other fungi.
So that's the first.
Yeah.
Because if I'm correct, all the previous work suggested only becomes endophytic in root tissue. So if you found it in the leaf,
yeah.
Well done.
Didn't know that, but yeah. Now I know.
Great, thank you very much. Okay.